see. The live stream always takes a few seconds to buffer.
Yeah.
Okay, still loading. All right, and it's going. Okay, so welcome everyone. It is my pleasure to introduce you to Charles Watson. He actually holds a doctorate in medicine and science. So he's an MD PhD and has a specialist qualification in public health medicine. He has published over 100 research articles and over 30 books. One of his books, A Red Brain Atlas, published with George Paxson, has over 100,000 times citation. So yeah, thank you, Charles, for being here. Thank you for your continuous support and involvement with the Australian Brain Bee as well as the International Brain Bee, and I'll hand the floor over to you.
Thank you, Marlene. First of all, I want to say to all of the students, congratulations on being international finalists. I know how much work it takes to get to this point when you've got other school studies at the same time in year 10. It's extraordinary how much people do. I mean, I know that these students know about as much neuroscience as a second-year medical student. It's an extraordinary amount they cover, not only in neuroanatomy, neurophysiology, but also in neurology. So congratulations and well done for the work that you've actually done to get here. My lecture tonight is entitled Mammalian Brain Evolution, but actually that's not really what it's about. Really what it's about is the fact that in neuroscience research and the kind of work that you do, nearly all the work that's reported is work that's done on humans or rats or mice. Very little involves other mammalian species, and certainly hardly anything involves non-mammalian species. So what I'm trying to do tonight is to tell you there are interesting things about other mammalian brains, and we're going to have a bit of a look through how you might investigate those yourself. So what I'll do is I'll try and load my PowerPoint presentation, and I can talk to that on the way through. Okay, so here it is. Sometimes I don't do this properly, but I'm trying. Is that? Let me just see. Oh, there it is. Okay, so I'll bring it up here. That's right. I'm hoping that you can all see mammalian brain evolution. Is that okay, Marlene?
Beautiful. Yes,
Okay.
awesome.
So just talking about mammals overall, the placental mammals, which are the main ones which are around the Earth at the moment, have been around for about 125 million years ago. But there are two other smaller groups that are significant. The egg-laying mammals, which have a lot of reptilian features. There are only two of them, the echidna and the platypus. They both live in Australia. And then, of course, there are the pouched animals, the marsupials in America and Australia. I'm, for various reasons, going to restrict myself to talking about the placentals tonight. And when you look about placental mammals, they're broken up into four main groups. And of these groups, two are relatively small, the Xenarthra and the Afrotheria. And there are two big ones, the Laurasiatheria and the Archontoglires, that are at the end. Let's look at the top two first. The Xenarthra include some unusual animals, the armadillos, the sloths, and the anteaters. They're in North and South America. The Afrotheria are another important but small groups. They include the elephants, the aardvarks, the tenrecs, and some other interesting ones. We're going to see an example of one tonight. And well, it's not today for you, but in this lecture. And then what I want to point out is that of the others, the Laurasiatheria, they include the cows, the hoofed animals, the whales, the shrews, and the bats. The important thing about these is that the bats aren't just any old another group. The bats constitute, as I said below, 25% of all mammals in the world. That's both in terms of the number of species and the actual number of animals. But we don't realize this normally because you don't see them much. They hide in caves and they're only around at night, so we don't notice the bats being so important. And to some extent, it's the same in the Euarchontoglires because the rodents here constitute 50% of all of the mammals on Earth. That's in terms of the number of species or the actual number of animals. But we don't see such a lot of them because they keep out of sight. Their success as an animal is that they avoid predators by staying hidden or underground, and they're very good at moving around at night time as well. So, of course, from a human point of view, the interesting thing about the Euarchontoglires is that they do include the primates and their relatives, the tree shrews and the flying lemurs. So, what I want to particularly emphasize tonight is the way of learning, appreciating more about mammalian brain evolution by making what I call a mini atlas. And the reason is that if you can get a set of sections of nice sections of an interesting mammal, you only need about 15 of them to make a mini atlas, and you need about 60 or 70 different labels. And the way that I've done this and other students have done this is to download them from the website called brainmuseum.org. It's a collection of 100 mammalian brains with sections from all of them, and the photographs were taken in the 1990s, so that we could have had better quality ones if they'd been taken only the last couple of years, but they're nonetheless pretty good, and they're a very interesting collection. I wanted to say a bit about brainmuseum.org. I want to do this partly because I think a lot of people have ignored the importance of brainmuseum.org over a long period, and I'll explain why I think that's true a bit later. But it's the only available collection of sections from the brains of many mammals. There are about 100 on it. And in most cases, the section sets were built at the University of Wisconsin in Madison under the supervision of Jersey Rose and later on Wally Welker. No, they're no longer surviving, and neither is John Johnson, who was a postdoc working with Wally Welker. He moved to Michigan State University sometime in the late 60s, and he decided to put together this collection of images to create brainmuseum.org. If you want to do one yourself, it's not awfully hard. You open brainmuseum.org, click on the heading saying brain sections, and there it will offer you 100 different mammalian species, and you pick on one that you like. I'm going to show you later on. When I was working with a student from India earlier this year, she picked on the armadillo, the 9-banded armadillo, and made a mini atlas of that. But you can pick whichever one you like. There's no reason for going in any particular direction. And when you look at when you click on the animal species, you'll see in brainmuseum.org there are some pictures of the animal, and there's a little bit of background about the characteristic behaviors of the animal. But you click on the thing saying Cell Picture Atlas, and then pick on the Nissel section ones and look at the ones there and select about 1 in 10 of the sections offered. About 15 sections is about enough. And you copy each section into a PowerPoint file and label them with standard abbreviations. Okay. I want to actually show you a quick example of one mini atlas. It's from a rodent, which is good because there are more rodents than anything else. And this is a quick atlas of the brain of the northern pocket gopher. And while I go through, I'm going to alert you to the sort of standard features of this animal, so that when you look at more unusual animals later on in the lecture, you'll realize how different they are. All of these sections were downloaded from brainmuseum.org and labeled. So we start at the front of the pocket gopher, and we just have the olfactory bulb with the olfactory ventricle. As we go back a bit, the cerebral neocortex starts to appear above that. Here's the olfactory bulb, and we can start to see some characteristic areas. The primary motor area is available first. The olfactory cortex, which replaces the olfactory bulb, is called the piriform cortex. We're going to see quite a bit of that. go a little bit further back, and we've got a lot more neocortex now, primary motor and primary somatosensory cortex. And below S1 is the lateral cortex of this group, the insular cortex, which is where taste is. And the Reinhold Fischer separates these neocortical areas from the olfactory piriform cortex below. And a new area, the olfactory tubercle, appears just underneath all of that. The ventricle starts to appear then. Go back a little bit further, and there's a lot more neocortex now. Once again, M1, S1, and now S2 as well, and the insula. And we can still see the piriform cortex and this rather wrinkly appearance, which is characteristic of the olfactory tubercle cortex. But now, a big lot of white matter underneath the cortex is about to cross the midline. That'll be the corpus callosum just there. And deep under the cortex are these bits of grey matter, generally referred to as the basal ganglia, but in this case it's the caudate and the putamen, and there's a ventricle in there as well. We go back a bit further, that we've got a lot more caudate and putamen, just the caudate there, putamen, and there are some speckly fibers streaking down between them, because some fibers from the cortex run from one cortical area to another. Some cross the midline as commissural fibers, and some go down to the brain stem, or some fibers come up from the thalamus up to the cortex. They're located in an area called the internal capsule. At the bottom of the caudate and putamen is an area which overlaps with them called the accumbens nucleus, just sitting down there. And lastly, on the medial side of the lateral ventricle is a big area called the septum. In the human brain, it's not so obvious. Still quite an important area, receives input from the hippocampus. And we're going to talk about that later. Once again, you can still see the piriform cortex and the crinkly olfactory cortex below underneath there. So we're now at a crucial section. The reason I say this is that when you look at brain sections, forebrain sections, the anterior commissure, while it's not as big as the corpus callosum, is a very, very good landmark. Behind the anterior commissure, we find the thalamus and the hypothalamus, and in front, we don't find those things. So it's very important to know this is the point behind which we're going to find the thalamus. But here we can see the caudate and the putamen and the internal capsule still between them. And a new thing has appeared. Underneath the putamen is an area which has a lot of white matter in it. So it's called the globus pallidus, situated just underneath that. There's another bit of the pallidum called the ventral pallidum, which is I've located just down there. Interestingly enough, at the very bottom, the second cranial nerve, the optic nerve, has appeared. Should be actually labeled 2 small n, but it's a mislabel just here, unfortunately. My fault. And we'll go into the next, of course. I do want to tell you something about the cortex. The motor cortex, you can sometimes see a line of dark cells in layer 3 or layer 5, which is characteristic of that. But in S1, characteristically, there's a nice layer of dark cells in layer 4, which we're seeing up and down there. We're going to talk more about that in relation to rats and mice, where they're much more interesting. And so we go to our next. So here, the thalamus has appeared in the middle. So we've still got the internal capsule, the caudate and the putamen. We've seemed to have lost the globus pallidus just here, but the thalamus with its various nuclei has appeared in the center there. We're not going to talk about the nuclei. Underneath that is the beginning of the hypothalamus. Here's the third ventricle, and the big dark nucleus is the suprachiasmatic. It's the nucleus which receives light input, and it's basically the 24-hour clock in the forebrain. Here's the optic chiasm, where the 2 optic nerves are crossing. Another interesting thing is that bulging into the ventricle at the top here is the extension of the hippocampus. The fimbria, the main fiber bundle leaving the hippocampus, is coming forwards to here to reach the septum and to go down into the hypothalamus. And we can see the beginning of the hippocampus. We'll see more of it in the next one. Yes, this next one has our classic hippocampal feature. And I want you to look carefully at this, because I'm going to show you some very different hippocampuses. I suppose they're hippocampi later on. But the features of the hippocampus that you should normally be looking for, there's a dentate gyrus, a bit like a sharp tooth just there. And then the corneomonas or CA regions coming out from that. And there's a modest size CA3. CA2 is too small to label just here. And most of the rest of the top here is CA1. We're going to see some examples with some very big CA1s in just a little while. Down in the thalamus, we've got all the ordinary things. Notable here is VPM, which is the touch nucleus that's appeared just down there. And down in the hypothalamus, the big ventromedial hypothalamus nucleus is on the side of the third ventricle. Interestingly enough, when we look at our cortex again over here, we've got S1, and I've got photographs in the road. I can't see what the rest of it is just there, but I'll hope that we'll find them all in a minute. But down in the temporal lobe, the amygdala has appeared. In the human brain, the amygdala appears at the front of the temporal lobe and is replaced quickly behind that by the hippocampus. Noted here that we had the optic chiasm in the last picture, and the optic tract is running here, going up towards the thalamus, where it will end. So here's our typical hippocampus above, dentate gyrus, with the CA regions, CA3, CA2, CA1, which is a bit bigger here than you might have expected in a mouse or a rat, is important because it's the spatial memory area of the hippocampus, and we're going to talk more about that with some of these other species in just a little while. In the meantime, though, in the thalamus, we've still got VPM, the touch cortex, and at the top we've got the dorsolateral geniculate, which is the visual area. And I can't see it quite here, but if we had a good series of sections, we would actually see the optic tract go up and end itself in the dorsolateral geniculate. The habenula is appearing on the medial side, and at the top we've got S2 and the other cortical areas. Interestingly enough, the ventricle, which was up the top here, winds down into the temporal lobe. It's one of these C-shaped structures, like the caudate and the fornix that go down to that, and sitting down under here. Next to that is the beginning of the hippocampus, the ventral part of the hippocampus, which is in the temporal lobe. In humans, nearly all the hippocampus essentially is located in the temporal lobe, whereas in most other mammals, most of it's in its original position, which it would have actually normally been connected with the neocortex, but the corpus callosum cuts the two of them off. So I'll quickly zip through the next three slides. Here, we've finally the central core has broken away from the rest of the forebrain, and we've got some thalamus with the dorsolateral geniculate, and underneath the mammillary bodies in the hippocampus. Here's the internal capsule I should have shown you last time, continues here as the cerebral peduncle. In the last section we were there, it was actually here, and I didn't point out to you. Oh, on this side we can see it, internal capsule going down into cerebral peduncle, just down below. The hippocampus at this point shows the continuity between the dorsal hippocampus and the ventral parts of the hippocampus as they are down underneath. This is a bit of subiculum just at the top there. The cortex has been replaced at the top by the visual cortex, V1, and then V2L and V2M are located apart from there. The third ventricle separates the two bits of the thalamus, and the posterior commissure, which has got to do with visual reflexes, is just seen there in the pretectal region. When we jump back a bit further, we're really into the brainstem. And the reason we know that is that the superior colliculus is just there. There's a bit of thalamus left over. The back end of the medial geniculate is just there, but the other brainstem things that are here are the cerebral peduncle, the substantia nigra, and the red nucleus, and of course we got with the aqueduct in the center. As we go further back, more brainstem things, superior colliculus and inferior colliculus, and then substantia nigra and cerebral peduncle down below, the interpeduncular fossa just there. This is the crossing of the superior cerebellar peduncle, which you may know about. And we'll jump quickly down to the last few. We're leaving the cortex behind now, and the cerebellum has appeared. So we've got inferior colliculus with bits of the cerebellum, cerebellar vermis in the center, And at the bottom, the pons has appeared. Of course, in the human brain, the pons is so expanded that it appears to cover the top half of the hindbrain, whereas in developmental terms, it really only starts from the second and third segments of the hindbrain, the second and third rhombomeres, quickly to the end, and we'll go on then. So here we're getting a little bit further. This is the principal trigeminal nucleus. We're going to see more of the trigeminal nucleus in a minute. The pons fibers have gone up here in the middle cerebellar peduncle, up towards the cerebellum. And in the next one, we can see more of the trigeminal system, the motor trigeminal, and the superior olive has appeared, the auditory nuclei with a trapezoid nucleus just down underneath. In the human brain, these are buried underneath the pons, so you don't actually often get a very good look at them. But the fifth, the motor nucleus of 5, you can see the big motor neurons on the side over there. As we go back a bit further, the seventh cranial nerve nucleus appears for the first time, although it'll be bigger later, and the vestibular nuclei appear at the top. And if we go to the next, we can see the very big motor neuron group of the facial nucleus. And of course, when you look at human sections, it's kind of buried above the main pontine crossing, so we don't get a very good look at it in human sections very well. And there are the dorsal cochlear nucleus is up the top. When we jump to the end, we'll finally get down to the inferior olive on the pyramid, and the ambiguous nucleus is just that little group in there with some vestibular nuclei up on top. So that's enough for a fairly standard mammalian brain. What I want to show you is some features of some different mammalian brains, ones that have specializations that are related to their sensory systems, because they're most obviously seen in these sort of sections. And these are the kinds of specializations that we can look for. Some animals have very big olfactory systems. The armadillos are one. I'm going to show you one of those. The tenrec is another. We're not going to look at that tonight. And of course, the primates and carnivores have very well-developed visual systems. Primates need them because characteristically they live in the trees and they need very good three-dimensional vision. And carnivores use them for hunting, most importantly. The carnivores also have pretty good auditory system for listening whatever is going on around them. Echolocation is an interesting sensation. We don't really have any idea about it ourselves, but bats have developed this incredibly. Not all bats are echolocators, but those that are can fly around at night and pick up insects from their echolocation that are flying around them and eat them. So that's one of the reasons why bats are one of the most successful groups of all the mammals. Interestingly enough, toothed whales have echolocation, and so do shrews. So it's not exclusive to the bats. Somatosensory elaborations are present in a few different animals. We're going to show you later on that rats and mice amongst the rodents have a fantastic somatosensory system devoted to their whiskers. Primates have pretty good somatosensory systems too, particularly for their touch. And raccoons, which are a carnivore, have wonderfully developed fingers with the somatosensory system. Interestingly enough, the specializations for the whiskers that were found in rodents have also been found in the marsupial, the brush-tailed possum in Australia. And finally, something I want to talk quite a bit about is that we have evidence of spatial awareness in a number of animals. The ungulates, the hoofed animals, spectacularly, pretty good in carnivores, but also in the capybara, this very big, huge, fat rodent, and the sengi. And we're going to show you some of these characteristics just now. So first of all, the armadillo. I said before, it's notable for its olfactory specialization. And armadillos eat ants and other insects, such as scarab beetles. They have a very keen sense of smell. And this is what their whole life relies on. But they can detect insects 6 inches underground. So they just go around, they can find insects wherever they are. And brain bee a student from Pune in India, Shatataraka Basarakati, has made a very nice mini atlas, and I'm going to show you one of the pictures from that now, of the armadillo brain. This is near the front of the armadillo brain, and the first thing you'll notice compared with the pocket gopher is that the amount of neocortex is not very great. Nearly the whole of its cortex is devoted to the olfactory areas, to the piriform cortex and to the olfactory tubercle underneath, as it swings around. So there's a little bit for the insula and the somatosensory cortex, but they don't have much room for the other major bits of neocortex that are there. Interestingly enough, this little thing that's labeled IG there is the hippocampal remnant, the indusium griseum, it's called, which you can see quite markedly in the armadillo, and that's the septum just underneath there. Okay, the red fox. Foxes are hunting animals. They of course have specialized visual and auditory systems. They have a spectacularly good, although not the biggest, CA1 hippocampal expansion for remembering the path of hunt. Because the problem for carnivores, if they hunt animals and they run around hunting them all day and night until they finally kill one, they've got to work out how to get home again. They usually vomit up some of the meat to feed their children when they get home. And I've got a picture here from a mini atlas made by Sam Richards, who's actually a Brain Bee finalist, which is terrific. Here's a picture near the front of the red fox brain. I'm going to show you a slightly enlarged one in a minute. The big features I want you to see is they've got a very good visual nucleus, the lateral geniculate in their thalamus, and they've got an interesting expansion in their hippocampus. I'll show you a bigger picture of it. You see, I showed you the hippocampus before, which had dentate gyrus and then CA3 and CA2, and CA1 was about that big. But look here, in the red fox, it's about 3 times bigger than you would expect it to be. I used to wonder about this until I saw a David Attenborough movie a few years ago of African wild dogs chasing a wildebeest, and these 10 wild dogs chased this wildebeest all until they finally brought it down. It took about 12 hours. And then, of course, they ate as much as they could. And I realized at that point that they would have to get home. They'd gone on a random all over the place, and they remember it very well. Of course, some of you will know that if you take dogs outside of a major city and dump them somewhere or another, they somehow or other manage to find their way back home by exactly the same process. They have very good spatial memory. Just to remark again, this is the very big lateral geniculate nucleus that the red fox is blessed with. Whiskers are very interesting. Rodents, obviously, since they're so successful, do extremely well with whisker tusk receptors. The thing about whiskers, they're not just hairs that stick out, but these are hairs which can be moved around almost like a searchlight, so that as they're walking along, if they're in the dark or in any area they're around, that they've got about half of the length of their body in front of them, this range of hairs that are feeling everything around there to go through all of that. And there are some neuronal concentrations in their cerebral cortex that are called barrels in the S1 cortex. It's an interesting thing. They're not very easy to see in standard sections, and they're actually only discovered. People have been looking at rat brains for about 100 years until Woolsey and Vanderloos found them in the 1960s. But one of the reasons is that you can really only see them properly in sagittal or tangential sections, and most people use coronal sections, so you don't see them very well. I'm going to show you what they look like in an ordinary section and then a special section later on, because these barrels are clumps of cells. There's one barrel for each whisker. I'll show you. So here's a standard rat brain picture from our rat brain atlas, and here's S1 with layer 4, and you can see a little bit here that there are some clumps. I think they're perhaps on this side over here, a little bit visible, but they wouldn't leap out at you at this, because you need a tangential section to see them properly. And I'm going to show you one of those in the next section. So here, This is a tangential section of mouse cortex to show you the way that these barrels are arranged. And amazingly enough, there's one row of barrel clumps for every row of whiskers. So this is whisker row A, B, C, D, and E, and each one of these has a row of barrels, and there's one barrel for each major whisker as you run along with that. This has been an incredibly important discovery because when people wanted to look at how the is organized developmentally, this is you know that every one of these barrels should be there under normal circumstances. So if you, for example, pluck out a couple of whiskers just after birth in a newborn mouse, and you look 2 weeks later, you find that 2 of the barrels are missing. So the barrel system has proved a very important topographical system for understanding the organization and development and recovery of the cortical areas. Okay, so this has really what got me involved in the business of looking at brainmuseum.org. I was watching about 8 years ago, sitting around, my wife was watching television on a Saturday night, and to entertain myself, I got out my computer and thought I'd have a little look at Brain Museum, and I saw they had a zebra brain. Now, zebras look very unusual, but they're basically just horses. They're exactly the same as all the other horses that are out there. But here's the extraordinary thing. When you look at a zebra brain, this is what struck me at the time. There's the dentate gyrus. Here's CA3 and CA2, and look at CA1. It just goes on forever. It's about 5 times as long as you would expect if it was a mouse or a rat or a similar sort of animal like that. It's hugely expanded, and this amazed me. What amazed me was that no one had ever noticed it before. It wasn't reported in the literature. And I realized later on the reason is that people like me who do neuroscience work on humans or rat or mice brains, and we never look at any other brains. And yet the veterinary students who look at horses and dogs, they never look at rat and mouse brains. So that there's a kind of a division between the people who might take on those things. So anyway, sorry, I just want to emphasize that the interesting thing about this is that because having found this, I thought, I wonder how many other animals have something similar. And when I went through all the mammals in brainmuseum.org, I found that all of the hoofed animals, the animals that like reindeer and deer and all sorts of other deer and things of that kind that live in herds, these are called the ungulate mammals. And of course, many of these animals conduct important annual migrations. They migrate to find better food at different times of the year. And some of the ones in Northern Europe migrate up to 2,000 kilometers. And it seems that the expanded CA1 enables them to remember where to go next year. The interesting thing is because they're herd animals, the young animals don't have to receive the information genetically. They learn the route from the older animals by traveling along with the older animals as they run along. So, as I said, this is what turned me on to what was so exciting about brainmuseum.org. I thought, I wonder how many other amazing things there are in there that people haven't noticed yet, and I hope that you might try and find some of those out later on. Here's just one more picture of the zebra brain to show you how wonderful it is. Here's our CA3, CA2, and here's all the CA1 as it winds around to give it that amazing spatial memory. Okay. Primates have spectacular visual specializations. The visual system is important because the primates characteristically live in trees, and they need precise 3-dimensional vision and well-developed motor skills to survive in that sort of environment. I haven't got such a great picture of a primate brain, but this is something from brainmuseum.org, not as well stained, but there's the lateral geniculate, and you can vaguely see here that it has different layers. So I'm going to show you one from the left-hand side in the next section to show you what these layers really look like. In primates, and in the higher primates, the lateral geniculate is actually organized into 6 quite distinct layers, and each one of them receives either from one eye or the other. For example, layer 6 receives from the opposite eye, layer 5 receives from the ipsilateral eye, but this organization is part of the story of a very highly developed visual system for three-dimensional organization. The raccoon I mentioned before is it's a carnivore, not all that different from a cat or a dog in many ways, but they have amazing hands. Instead of just having a paw, they have hands with separate fingers and flexible thumbs, and they're extremely good at opening containers of various kinds. They do particularly well with rubbish bins, and so they've flourished well in human settlements. They can basically get anything they want out of anything that's around in the backyard of a human settlement. And when you actually look at their somatosensory cortex, it's not such a great picture I'm going to show you, but it's a pretty good one. That you'll find that they have one gyrus for each hand digit. I mentioned before Wally Welker from Madison, Wisconsin, and John Johnson, and they're people that did the important part of this work. Oh, what have I done here? Oh, sorry. It's just to show you that this is a section through a raccoon brain, and these are these separate gyri. I can't prove to you which ones they are there, but the next section shows you that quite well. Here's a picture of the raccoon brain from the lateral side, and these are the areas that these people recorded from. They recorded the touch sensation, and they found that each digit had its own little gyrus, so that the 5 digits in the front paw are each represented by a special gyrus, and so that the rest of the somatosensory cortex is not that large. Here's the mouth and the face. That's the area just down underneath there, and then the arm and other areas are located, shoulder and arm a little bit down there as well, outside of that. But there's a disproportionate area that's devoted to the digits in the hand. The Sengi, I must say I didn't know about the Sengi until I watched the David Attenborough documentary about 10 years ago, and if you like looking at David Attenborough, it's worthwhile looking this one up. It's called the elephant shrew. It's an about the size of a small rodent. It lives in tall grass, and the interesting thing about the animals is they spend all day walking through the grass, clearing the path. They eat insects along the way, but their main thing is to make sure the paths are absolutely clear and safe for running along, and also that they remember every millimetre of every path. So when they're attacked by birds from above, they race along these paths at an incredible scene. You will enjoy the David Attenborough documentary if you can find that. There's one available on the web, and it's only about 15 seconds, but the speed with which they move through these paths is absolutely fantastic. Here's a Sengi. They do have a long snout, so they are related to the elephants, and so they show a tendency to have a snout like a part of their face, and this is what their brain looks like. When you cut a section through their forebrain, the hippocampus is so big that in volume, it's around about the same volume as the rest of the whole of the neocortex. It takes up an enormous part of their cortical structure. And once again, here's the dentate gyrus. Here's CA3, little tiny CA2, and CA2 once again for spatial memory is very much expanded, and that's their secret in terms of their ability to move fast along these paths and work out where they're going in the end. Just out of interest, this is a very nicely stained section. You can see many of the thalamic nuclei. You can see the paraventricular hypothalamic nucleus, the supraoptic nucleus. They're the ones which send the fibers down to the posterior pituitary. Show up really, really beautifully on this. And of course, they have a pretty good olfactory system too, but they're not primarily dependent upon that. I mentioned that echolocating bats are important. Bats are the only true flying mammals. So some mammals can float from one tree to another with a wing-like structure, but they're not really flyers. Bats are the only ones, and of course they detect flying insects at night with echolocation. They do have a lot of specializations in their auditory system, but from an anatomical point of view, they're not quite as spectacular as perhaps the CA system in the Sengi or in the zebra. But I'm going to show you a picture nonetheless. Here's a picture of their brainstem. The infraoculus is certainly larger than you would expect for an animal of that size. And you can see at the top, perhaps even better, here's the supraoculus. The infraoculus is bulged right up above there. And they have a lot of specializations in their superior olive, which seem related to their ability to detect the three-dimensional nature of the echolocation. And I think the last animal I'm going to show you is an extraordinary one. Once again, a student that I was involved with made a mini atlas of this. The capybara is by far the largest rodent. They weigh averagely about 50 kilograms, but they can go up to 80 kilograms. They're as big as a huge pig. And they're entirely herbivorous. They eat grasses and aquatic plants. They spend quite a lot of time in the water. They roam over a quite large range, which I think is important to what I'm going to show you in a minute, to find their preferred their preferred foods. That they can extend over about 25 acres. They normally live in groups, about 25 or more. And they do have a very gyrated neocortex. When you're used to looking at mouse or rat brains, there's not much going on in terms of folding of the cortex, but these ones have that. And if you look underneath the cortex in the hippocampus, you find they've got expanded spatial memory capability. Here's a capybara, a medium-sized capybara. And here's what their brain looks like. It's a very gyrated cortex for a rodent. They don't have much in the way of sense of smell because they're basically just looking to find plants which are sitting on top of the land. So they don't smell them out. They look for them to find them. But look at their hippocampus once again. See, the dentate gyrus. Here's CA3, and here's you find in carnivores, not quite as big as horses or ungulates, but very, 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 very nearly. I've wondered about how exactly they use this, and I can only imagine that when you patrol a 25-acre area, there must be good things to eat in some places, and it's good to remember where they always come up. And so they somehow or other have a capacity to go and find the good eating areas on a regular basis because they obviously eat pretty well, given how they look. Okay, so what can you do? I'd be thrilled if you had a look at brainmuseum.org when you've got time and consider making a mini atlas of the brain of a single placental mammal. If you find one and you make it and you're struggling through it at all, you're welcome to send it to me and I'll do corrections on it and see if you can straighten that out a little bit. I strongly recommend using a standard set of abbreviations. It always annoys me when I get articles to review and people make up their own abbreviations. George Paxanos and I made up a very good abbreviation set in 1982, and it's the most widely used in the world now. And it's a good idea to use a standard abbreviation set because it saves everyone a lot of trouble. That's all I have to say tonight. Thanks for listening. How's that, Marlene? Is that all right?
Awesome. Thank you so much. That was very wonderful, very inspiring. Fun to
Thank
see
you.
so many different brains. Are there any questions from the audience?
Please.
I have a bit of
Someone's.
a question I was wondering about because I was wondering because humans have very dexterous fingers. Do we have a similar cortical thing to raccoons?
No, we don't. It's a very good question actually, because given how much we rely on our fingers, we have the whole of our first somatosensory area has big areas for them, but they're not separate little gyri the way that you would see in raccoons. Raccoons are very, very special. But it does point out something else, and that is that just because different animals evolve a particular sensory specialization, it doesn't mean that they'll all handle it the same way. They may have different ways of coding the stuff that does things. So yeah, you're right. I mean, you might have hoped that you'd be able to find one gyrus for each finger, but you actually don't. And yet we obviously have fantastic sensation in our fingers. Other questions?
Awesome. I think there's a question from Gina in the chat that I can read out. Thank you so much for this wonderful lecture. You really opened my eyes with all of this diversity in the mammalian brain. I was wondering if certain brain areas would be larger in mammals that live in groups. Is it very visible that certain animals are more social than others, aside from generally having a larger neocortex?
I don't know the answer, unfortunately, but it's a very good question, actually. We noted a moment ago that the capybara, one thing that's notable about that is they live in quite big family groups. And of course, all of the hoofed animals live in herds. They live in very, very big groups. And maybe if you live in a group, the group can learn how to get around a particular area, whether it's a migration route or where to find the food, and the information can then be passed on to young animals while they're growing up and they move with the group. But I've never thought of it before as just groupness as being a particular thing. But I'll certainly look more for that. Thanks for the suggestion. Great idea.
Awesome. Look at you doing research, Gina. This is amazing.
Yes.
Nice. Are there any other questions from the group? Okay. I guess it was very clear. Thank you so much, Charles. Please all join us in a round of applause for Charles. Thank you so much for your
Thank you.
time.